Hello, my name is Norhana Jane P. Kanakan. Today I will be talking about the Introduction to Applied Bacteriology. Introduction to Applied Bacteriology. So, before we start, what is bacteriology? Bacteriology is the branch of microbiology that deals with the morphology, structure, classification, and biochemistry of bacteria. So, the principle of bacteriology arose during the 19th century from scientific attempts to prove the germ theory of disease, namely that diseases were caused by microscopic organisms invading host cells. Bacteria are prokaryotic single microorganisms that are metabolically active and divides by binary fission. Some of these organisms play a significant role in the pathogenesis of diseases. Management of bacterial diseases is generally with antibiotics. However, the choice of antibiotics may vary depending on bacterial structure and metabolism. What is applied bacteriology? Applied bacteriology is the application of bacteria in the aspect of biotechnology for the production of new medicines, vaccines, biotechnologically engineered products for beneficial of humans and animals. Pure bacteriology deals with the profound theoretical study of the organisms as the subject itself. In comparison, applied bacteriology or biotechnology deals with the application of microorganisms in specific process such as brewing or fermentation. Bacteriologic analysis of water. Bacteriologic water testing is a method of collecting water samples and analyzing those samples to estimate the numbers of bacteria present. This note presents the background to the testing of water samples to determine whether disease causing bacteria, in particular fecal coliforms, are present in the water. Here are some of the bacteria that are usually found: the Aeromonas hydrophilia, Enterobacter aerogenes, Enterococcus species, Escherichia coli, Klebsiella species, Listeria species, Pseudomonas aeruginosa, Salmonella species, as well as the Shigella species. As much as I'd love to discuss this further, this was assigned to my other group mates. So back the bacteriological analysis of milk. Certain bacteria convert milk into useful products such as buttermilk, yogurt, and cheese. Commercially, cultured buttermilk is prepared from milk inoculated with a starter culture of the Lactococcus, usually Lactococcus lactis or the Lactococcus lactis cremoris. So it's important to examine milk for pathogens to ensure that it's safe to be consumed by humans. It's also considered as an hygienic determinant. Here are examples of bacteria found in milk: the Streptococcus lactis, Achromobacter, Bacillus subtilis, and the coliform bacteria. Thank you for listening.
Hello everyone, I am Vanessa Talatala and I'm going to report about bacteriologic analysis of water, specifically about the characteristic of bacteria in water. So for the introduction, water undoubtedly one of the most precious resources on our planet, sustain life, supports ecosystem and is essential for human survival. However, not all water sources are safe to drink or use without proper treatment. So microorganisms that cause disease by transmission through contaminated water are called waterborne pathogen. So bacteriologic examination is a comprehensive process used to assess the microbial quality of water. So one of the main job of water system operators is to control potentially pathogenic microorganisms. So the primary goal is to identify potential pathogen such as Escherichia coli or E. coli, Salmonella, and Vibrio cholerae which can cause waterborne diseases like cholera, dysentery, and gastroenteritis. So next is the general characteristics of waterborne pathogen or the bacteria that live in water. So for the size range, so we have zero point 0.5 to 2 microns in diameter, so 1 to 10 microns in length of the bacteria. So another way to visualize the size of the bacteria is to consider that it would take about 1,000 bacteria lying side by side to reach across the head of the straight beam. So there are three general groups of bacteria based on their physical shapes. So we have three shapes. So rod-shaped bacteria, these are called bacilli. Spherical-shaped bacteria, these are called cocci. We have spiral-shaped bacteria, which make up the third group. So within this group, there are many different arrangements. Some exist as single cell and others as pairs, as packet of 4 and 8, and as chain or as clumps. So next is the food requirement. So the food requirement, most bacteria require organic food in order to survive and multiply. So the food comes from plants and animal material that gets into water where bacteria exist. So some bacteria can use inorganic food such as iron as an energy source. So the bacteria convert the food into energy and use the energy to make new cells. So these bacteria can exist and multiply even when the organic pollution is not present. So for the temperature requirement, the bacteria will multiply more quickly when it is warm, and more chlorine may be available to obtain proper disinfection. Typically, for each increase of 10 degrees Celsius, the growth rate doubles. So, the temperature affects the rate of which the bacteria grow. So, the warmer the environment, the faster the rate of the growth. So next is the pH requirements. So pH also affects the growth of the bacteria. So most bacteria grow best at neutral pH. So extreme acidic or basic condition will inhibit growth, including some material such as metal ions and some organics such as pesticides and herbicides, which are toxic for bacterial growth. So next is the oxygen requirement. So oxygen requirements, we have three. So mainly bacteria are aerobic. So aerobic means they are require free or dissolved oxygen in their aquatic environment. So few bacteria are anaerobic. So when we say anaerobic means they can exist and multiply in the environment that lacks the oxygen. So some bacteria that are normally aerobic can switch to anaerobic, so which are called facultative. So one of the most troublesome bacteria in the water business is the iron bacteria, which is facultative organisms. But the bacteria responsible for biological treatment of wastewater are aerobic. So next is on how the bacteria multiply. So under optimum condition, bacteria grow very rapidly. They multiply by simple dividing process, which referred to as binary fission. So bacteria growing under optimal condition can double their number for about every 20 to 30 minutes. So this means that as long as the nutrient hold out, even the smallest contamination can result in sizable growth in a very short time. So these are the common bacterial waterborne diseases. So we have Salmonella typhi, Shigella species, Vibrio cholerae, Campylobacter species, Enteropathogenic E. coli, and Leptospira species. So that's all. Thank you for listening.
Good day, everyone. I am Althea Bell Tipira. Today I'm going to discuss the microscopic examination of bacteriologic analysis of water. Microscopic examination of waterborne bacteria involves staining, observation of cellular morphology, and quantification to assess the microbial quality of water samples. Different staining techniques and additional confirmation tests may be required depending on the specific bacteria being examined. So the following slides are the different types of bacteria under the microscope and that can be found in water. So, Escherichia coli is one of the bacteria that is commonly found in water. E. coli cells are typically stained using gram staining, where they appear as gram-negative rods, and they are facultative anaerobes non-sporulating coliform bacterium, and under the microscope, cells are typically rod-shaped and are about 2.0 micrometer long and 0.25 to 1 micrometer in diameter, with a cell volume of zero point 0.6 to zero point 0.7. These bacteria are often fecal in origin and can be harmful to human health. Though most E. coli bacteria are harmless and are found in great quantities in the intestine of people and warm-blooded animals, some strains, however, can cause illness. E. coli in water is a strong indicator of sewage or animal waste contamination. Sewage and animal waste can contain many types of diseases causing organisms. Consumption may result in having gastrointestinal infections, diarrhea, abdominal cramps, hemorrhagic colitis, urinary tract infections, and in severe case is kidney failure. Next is Salmonella. Salmonella can be stained using gram staining, but they are often detected using specific antibody-based staining techniques. Under the microscope, they appear as gram-negative rod-shaped bacteria. Generally, they measure 2 to 5 microns long by zero point 0.1 to 1.5 microns wide and motile by peritrichous flagella. Salmonella is a bacterial resident found in the intestinal tracts of many species, ranging from birds and reptiles to livestock, and can also cause bacterial disease that affects the intestinal tract of human. Water containing salmonella can come from a variety of sources, including agricultural runoff, sewage leaks, or stormwater runoff. Wells can also become contaminated by salmonella after flood or other natural disasters. When salmonella is consumed by humans through contaminated water, it can result in salmonellosis, a condition characterized by fever, severe intestinal distress, diarrhea, and vomiting. Next slide is Vibrio cholerae. Vibrio cholerae can be stained using gram staining, but they are often detected using specific staining techniques or molecular methods. Under the microscope, Vibrio cholera is short, curved, comma-shaped, gram-negative bacilli. They measure about 1.5 microns to 0.2 to 0.4 microns in size. They are rounded or slightly pointed ends, and they are a natural inhabitant of aquatic ecosystems worldwide. So, typically residing in coastal or brackish water. Vibrio cholera in water indicates a fecal contamination. Consumption of water with cholera can cause a person to have a cholera. So this infection is often mild or without symptoms but can be severe. Approximately 1 in 10 people who get sick with cholera will develop severe symptoms such as watery, diarrhea, vomiting, and leg cramps. Rapid loss of body fluids leads to dehydration and shock. Without treatment, death can occur within hours. Next is Shigella. Shigella cells can be stained using gram staining or specific antibody-based staining techniques. Under the microscope, they appear as gram-negative, non-motile, facultatively anaerobic, non-performing rods. Their cells are 0.4 to zero point 0.6 micrometer across by 1 to 3 micrometer long, and they are normal inhabitants of human intestinal tract and can cause shigellosis through consumption of water that has come in contact with the feces of an infected person. And it will develop symptoms such as abdominal pain, tenesmus, water diarrhea, uh, watery diarrhea, or dysentery, which multiple scanty bloody mucoid stools, and other signs may include abdominal tenderness, fever, vomiting, dehydration, and convulsions. Campylobacter are often difficult to stain using conventional staining methods or molecular methods may be used for detection. Under the microscope, Campylobacter appears as gram-negative, non-performing. They form motile, spiral-shaped rods that are zero point 0.2 to zero point 0.9 micrometer wide and zero point 0.5 to 5 micrometer long, and it moves by a corkscrew-like motion. So water contaminated with Campylobacter is known to result from septic seepage and wastewater intrusion into non-disinfected sources of groundwater, and occasionally from cross-connection into drinking water distribution systems. Consumption of contaminated drinking water is a significant cause of campylobacteriosis, which leads to diarrhea, abdominal pain, and cramps. Fever and vomiting and symptoms usually begin within 2 to 5 days after exposure to organism. Legionella pneumophila is a worldwide waterborne and most common cause of cases including outbreaks are commonly found in lakes, rivers, creeks, hot springs, and other bodies of water. Inhalation of contaminated aerosols from contaminated water may cause a Legionnaire's disease, which has an initially symptoms of fever, loss of appetite, headache, malaise, lethargy, and some patients may also have muscle pain, diarrhea, and confusion. There is also usually an initial mild cough, but as many as 50% of patients can present phlegm, blood, streaked phlegm, or hemoptysis occurs in about one-third of the patient. The severity of disease ranges from a mild cough to rapidly fatal pneumonia. Death occurs through progressive pneumonia with respiratory failure and or shock and multi-organ failure. Legionella pneumophila can be stained using specific fluorescent antibody techniques or other specialized staining methods. Under the microscope, it appears as rod-shaped bacteria. Cells are thin, somewhat pleomorphic, gram-negative bacilli that measure 2 to 20 micrometer. Enterococcus fecalis is an important opportunistic pathogen which is frequently detected in mineral water and spring water for human consumption, and it causes human urinary tract infections, endocarditis, and neonatal sepsis. Enterococcus fecalis can be stained using gram staining, where it appears under the microscope as gram-positive small cocci in clusters, short chains in diplococci, and non-sporulated and non-capsulated, and it measures zero point 0.5 to 1 micrometer in diameter. Next is Mycobacterium avium, a waterborne opportunistic pathogen whose characteristic makes urban water distribution system and household plumbing ideal habitats for their survival, persistence, and growth. Rather than contaminants, Mycobacterium avium are colonies of drinking water system. It causes pulmonary bloodstream and skin infections in susceptible individuals. Mycobacterium avium is an acid-fast bacterium, meaning it retains certain dyes despite decoloration with acid alcohol. So acid-fast staining techniques such as Ziehl-Neelsen or Oramine rhodamine staining are used. Mycobacterium avium cells appear as slender, rod-shaped bacteria under the microscope, often forming clumps or aggregates, and they measure 2 to 5 microns long by zero point 0.5 to 1 microns wide. And that's all. Thank you for listening.
So, hi everyone. My topic is about Methods for analysis. So analysis for bacteriologic analysis of water. So what is bacteriologic analysis of water? This for analyzing or and also estimate the numbers of bacteria present and to find out what sort of bacteria they are. It's like gram-positive bacilla, gram-negative, or non-swarming, or swarming, non-motile or non-motile. It represents one aspect of water quality. How this method helps. It is a microbial analytical procedure which uses samples of water and from these samples determines the concentration of bacteria. So if you have a sample and you will determine the concentration of bacteria through these methods and possible to draw inferences about the suitability of the water for use from this concentration. So methods used in culturing of water. So analysis usually performed using cultures, biochemical tests, and sometimes optical methods. But these methods is depends on what kind of bacteria is present. So of course, if you already know what types of bacteria is, you should put them to the methods that will enhance their growth. So you will not wasting your time. You will not waste your time using not suitable methods. So that's it. So these methods used for analysis of water. So the first one is multiple tube method, methodology of bacterial analysis of water, membrane filtration, for plate method, and plate count. So multiple tube method, one of the oldest method, is called a multiple tube method. In this method, a measured samples, perhaps 10 ml, is diluted with 100 ml of sterile growth medium and aliquot of 10 ml is then decanted into each of 10 tubes. The remaining 10 ml is diluted again, and the process repeated. At the end of 5 dilution, this produce 50 tubes covering the dilution range of 1:10 to through 1:1000. So methodology of bacterial analysis of water. The tubes are then incubated at preset temperature for a specified time, and at the end of the process, the number of the tubes with growth in is counted for each dilution. Statistical tables are then used to derive the concentration of organism in the original sample. This method can be enhanced by using indicator medium which changes color when acid-forming species is present and by including a tiny inverted tube called Durham. Durham tube in each sample's tube it catches any gas produced. This production of gas at 37 degrees Celsius is a strong indication of the presence of E. coli. So membrane filtration. Most modern laboratories use a refinement of total plate in which serial dilution of the samples are vacuum filtered through purpose made membrane filters and these filters are themselves lying on nutrient medium within sealed plates. The methodology is otherwise similar to conventional total plate counts. Membrane have printed millimeter grid printed on and can be reliably used to count the number of colonies under binocular microscope. For plate, when analysis looking for bacterial species that grow poorly in the air, the initial analysis done by mixing serial dilution of the sample in the liquid nutrient agar, which is then poured into bottles which are then sealed and on their slides to produce a sloping agar surface. Colonies that develop in the body of the medium can be counted by eye after incubation. The total number of colonies is referred to as the total viable count or TVC. The unit of measurement is CFU ml or colony forming units. per milliliter and relates to the original samples. Calculation of this is multiple of the counted number of colonies multiplied by the dilution used. And pour plate method, test of contamination in which the number of the colonies of coliform bacteria Escherichia coli or E. coli per 100 milliliter of water is counted. The result is expressed as coliform microbial density and indicates the extent of fecal matter present in it. According to common water quality standards, drinking water must be completely free from any colony. Bathing and swimming pool water can have about 200 colonies, and recreational fishing and boating water about 1,000 colonies. So that's all, and thank you for your listening.
Good morning, Sir Kent. I am Omar Jahir DR, and this is the bacteriologic analysis of water and the interpretation of result. In interpreting the result of bacteriologic analysis of water, it involves the understanding the presence and the concentration of different bacteria and microorganisms in the sample. One of the example is coliform bacteria. Coliform bacteria are group of bacteria is commonly found in the environment and in the intestine of warm-blooded animals. It also presence indicates possible fecal contamination and the potential of presence of other harmful pathogens. The interpretation usually involves comparing the number of coliform colonies found to establish regulatory limits. One of the example of this, sir, is in drinking water. If there's a presence of coliform bacteria should be absent or below a certain threshold. Just like they said before, sir, is a one colony for forming unit is equivalent of unit per 100 milliliters of water. And Escherichia coli or E. coli is a specific type of coliform bacteria that is found in the intestine of humans and animals. And also its presence in the water is a strong indicator of recent fecal contamination and potential presence of other pathogens, similar to coliform bacteria. The interpretation involves comparing the number of E. coli colonies that found to regulatory limits. And next is the total plate count or the TPC. This is the test that measure the total number of viable bacteria present in the water sample. High TPC, it could be indicate the poor water quality and the potential of presence of pathogens. While the low TPC, it could be indicate a good quality of water quality and it not be a potential of presence of pathogen. Next is the presence of specific pathogens. Just like what I said before, one of the specific pathogen present is the water is the Escherichia coli. And also remember it also Salmonella, Shigella, Vibrio cholerae, and also the viruses such as norovirus or rotavirus. And I think that's all for today, sir, and thank you.
Hello everyone, I am Lovely Faith Rose Reyes Camacho and I am reporting to you the pros and cons of bacteriological analysis of water. So first, what is bacteriological analysis of water? It is a method of collecting water samples and analyzing those samples to estimate the numbers of bacteria present. It is able to detect the presence of harmful microorganisms such as bacteria. So first, para makabalita kung anong naa ang naay mga bacteria sa water, we have to recall what is the cycle of water. How does bacteria get into the water? So this is the water cycle. Of course, familiar tang tanan unsa ang water cycle. So condensation, precipitation, and infiltration to evaporation again at to condensation. Ang evapotranspiration, mauni siya ang water movement from soil to atmosphere via plants. So as you can see, i-take up sa plants ang water sa ground. Then, mag-evapotranspire siya, mag-condensate from the sea, mag-evaporate siya kung madtong kung init kaayo, kay mag-taod-taod mag-ulan, kay of course, from evaporation, the cloud will get heavy, then it will cause rain. Then, pag mag-rain sa ground, then i-infiltrate niya, then of course, back na po sa atong large bodies of water such as seas and rivers. Next is this diagram. Mao ni dili to makita kung asa unsa-unsang bacteria ang gikan sa to industrial waste, wastewater treatment drain, animal farm feces, domestic animal feces, and of course, household sewage drains such as Vibrio cholerae, Escherichia coli, Salmonella typhi, Pseudomonas aeruginosa. Ang culture-dependent methods, these are the methods on how they make the water safe for drinking. These are heterotrophic plate count, most probable number method, membrane filtration method, defined substrate enzyme method. Ang heterotrophic plate count is a method that measures colony formation on culture media of heterotrophic bacteria in drinking water. Bacteria such as E. coli, Salmonella, Shigella, mo ni siyang usually makita. So most probable number method, it is a statistical method used to estimate the number of viable cells of a particular microorganism in sample, most commonly in food and water. A membrane filtration method utilizes a selectively permeable membrane to separate oligomers of different molecular sizes. So this way, mas differentiate ang bigger sizes ng dirt sa drinking water and of course bacteria. Define substrate or enzyme methods. In some reaction, one substrate is broken down into multiple products. In others, two substrates come together to create one larger molecule to swap pieces. So this diagram will represent the culture-dependent methods. Water-contaminated bacteria, of course, must screen siya through cultural-dependent methods. Here are the pros of bacteriological analysis of water. First is identifying pathogens, public health protection, early warning system, compliance monitoring, quality control, and research and development. Identifying pathogens. Pathogens such as E. coli, Salmonella, Vibrio cholerae, Shigella, Campylobacter, Legionella pneumophila, Pseudomonas aeruginosa, and E. faecalis, Clostridium perfringens are some of the bacteria, and identifying pathogens would help to identify pathogenic bacteria that can cause waterborne diseases like cholera, typhoid fever, and gastroenteritis. Public health protection, safeguarding public health by ensuring that drinking water meets safety standards. So, this way, ma-ensure na kina-drink nato mostly kinaghan nagainom through tap water is of course consumable by us humans. Early warning system allowing authorities to detect contamination issues promptly and take corrective actions to prevent widespread outbreaks. So, through this early warning system, mas madali ma-announce na, for example, ang water is contaminated with bacteria that will cause diarrhea and vomiting, and of course, stomach aches commonly. Compliance monitoring ensure compliance with health and safety regulations for quality control, monitoring the effectiveness of treatment processes, and identifying any potential sources of contamination. By QC testing for harmful pathogens such as E. coli and coliform bacteria, water treatment facilities can ensure that drinking water is safe. So mostly sa tropical areas, ang makita is Vibrio cholerae. In the temperate zone would be Salmonella typhi and Salmonella typhimurium. Research and development, aiding in the development of improved water treatment methods and strategies for preventing waterborne diseases. So here are the bacteria sa water and the disease they cause. So Escherichia coli, Salmonella, Vibrio cholerae, Shigella, Campylobacter, Legionella pneumophila, Pseudomonas aeruginosa, Enterococcus faecalis, and Clostridium perfringens. So as you can see, mostly ani is gonna cause bacteria, abdominal cramps. Of course, in immunocompromised individuals, it will lead to UTI and other healthcare associated infections, but mostly diarrheal disease, stomach cramps, and fever, of course, minor illnesses. The cons of bacteriologic analysis of water, it would be time consuming, cost, complexity, limited scope, false negative, positive sampling techniques. Time consuming, of course, it will require several days for culturing bacteria and obtaining accurate results, which will take time para came up sa answer. So, cost, the equipment, reagent, of course, they will use money and funds to provide for the equipment needed, reagent for the test, and of course, in the future, persons like us na mag-test o water. Complexity involves complex laboratory techniques and procedures requiring trained microbiologists and technicians to perform accurately. Sa limited scope, it will focus on detecting bacteria and may not identify other contaminants such as chemicals, heavy metals, or parasites that can pose health risk in water. Mas challenging siya sa large and complex body networks of water. And cross connection sa backflow may cause improper plumbing connections or the pressure changes which will cause the possible bacteria to divert in other water channels that will cause bigger mass or damage. The false negatives or false positives, there is a possibility of false negatives or false positives, which can lead to misinterpretation of water quality and potential health risk. So, ang pesticide, fertilizer, and livestock waste will also cause fecal contaminations. Sampling challenges crucial for accurate bacteriological analysis, but obtaining representative samples from different points in a water distribution can be challenging. Then again, large and complex body networks of water will be challenging for this bacteriologic analysis of water. However, bacteriological analysis of water will play a vital role in protecting public health and preventing waterborne diseases. Again, I am Lovey Faith Rose Camacho, MLS 3C. Thank you for listening.
Good day everyone, my name is Janvela Seblos, and for today I will be discussing about the characteristic of milk in a bacteria. Microbiology of milk. The composition of milk, it contains more than water than other elements, around 87% for dairy cows. Under the microscope, cream can be seen to consist of large numbers of spheres of varying size floating in the milk. Each sphere is surrounded by a thick skin which act as the emulsifying agent for the fat suspended in milk. The quantities of the main constituent can vary considerably depending on the individual's animal, its breeding, stage of lactation, age, and health status, herd management practices, and environmental condition. So in this, milk provides the ideal environment for bacterial growth due to its nutritional components. Dairy Dairy microbiology encompasses a broad range of bacteria, which some may cause disease spoilage, which others are intentionally added for fermentation in products like yogurt and cheese. By understanding the milk microbiology, researchers and practitioners can implement strategies to ensure the safety quality of the dairy product. Next, we have the composition of a cow's milk. So the main constituents are water with a range of 85.5 to 89.5. Total solids are 10.5 to 14.5. Fats is 2.5 to 6. Proteins is 2.9 to 5. Lactose is 3.6 to 5.5, and minerals is 0.6 to 0.9. So microbes can enter the milk via cow, air, feedstuffs, milk handling equipment, and the milker. Once microorganisms get into the milk, their numbers increase rapidly. It is more effective to exclude microorganisms than to control microbial growth once they have entered the milk. Milking equipment should be washed thoroughly before and after use. Rinsing is not just enough. Bacterial types commonly associated with the following: Pseudomonas for spoilage, Brucella for pathogenic, Enterobacteriaceae is pathogenic and spoilage, Staphylococcus aureus and pathogenic, Streptococcus S. agalactiae is pathogenic, S. thermophilus acid fermentation, S. lactis acid fermentation, same goes by the S. ramonis and S. lactis diacetylactic. Leuconostoc lactis is acid fermentation, Bacillus cereus is spoilage, Lactobacillus, L. lactis, L. bulgariceus, L. acidophilus, and Propionibacterium are acid production, and Mycobacterium tuberculosis is pathogenic. So the major milk-borne pathogen and their associated disease are: first, we have the Campylobacter jejuni, which the disease is gastroenteritis. Disease symptoms are diarrhea, abdominal pain, fever. Source are from the intestinal tract and feces. So this bacterium is prevalent in the intestine and feces of cattle, poultry, and wild birds, as well as in contaminated water. It is a common cause of diarrheal illness, and symptoms include what I have said earlier and last 5 to 10 days, and can lead to more serious condition in individual with compromised immune system. Next, we have the Coxella brunetti. This is a Q fever. It has a disease symptoms with chills, fever, weakness, headaches, and possible endocarditis. Source is from infected cattle, sheep, and goats. Found in animals like cattle, goats, sheep. It can also cause Q fever in humans. Symptoms include what I have said earlier, and it can lead to death and severe cases. So next we have the Escherichia coli 0157 or the H7. This strain of E. coli is found in the intestines and feces of a cattle. It produces toxins that cause symptoms such as a bloody diarrhea, abdominal cramps, and severe cases. It also hemolytic uremic syndrome or the HUS, which could result in kidney damage or failure. Pasteurization could kill or could destroy the E. coli 0157H7. Next, we have the Listeria monocytogenes, widely found in soil and water. Listeria monocytogen can cause listerioid, particularly dangerous for pregnant individuals, newborns, and elderly, and those with weakened immune system. Symptoms including flu symptoms, fever, muscle aches, and gastrointestinal issue can also lead to more severe conditions such as septicemia and meningitis. Next, we have the Mycobacterium bovis or the tuberculosis. Both bacteria are destroyed by pasteurization and found in infected cattle. They could cause tuberculosis primarily in lungs disease, but historically associated with milk consumption. Next, we have the Mycobacterium paratuberculosis or the Jones ruminants disease in cattle and has been suggested as the potential link to Crohn's disease in humans. Although this is link is not confirmed, this our source is infected animals. Next, we have the Salmonella species or the gastroenteritis typhoid fever. Various strains of Salmonella causes illness to humans with um with the symptoms including diarrhea, abdominal cramps, and fever. In severe cases, it could lead to hospitalization. Some individuals may develop reactive arthritis as complication. And lastly, we have the Yersinia enterocolitica that is foundly seen in intestinal tract of farms animals, especially pigs, and in the environment. Yersinia enterolytica can cause fever, abdominal pain, and diarrhea in humans. It can grow in food products are contaminated after pasteurization. Um, Next, we have microbial safety in handling the milk. All fresh fluid milk should be stored at a temperature below 40 degrees Fahrenheit and should not be stacked high in display cases. Milk that can purchase should be taken from the store and quickly placed in your refrigerator at home so that the temperature does not rise above 40 degrees Fahrenheit. Pasteurization, wherein milk is heated so that the temperature is sufficient to kill the bacteria. Pasteurization heats milk to kill harmful bacteria, extend its life shelf. Heat milk to 72 degrees Celsius for 15 seconds, followed by a rapid cooling. Batch pasteurization used for smaller quantities, heat to 63 degrees Celsius for 30 minutes. While pasteurization does not significantly alter milk nutritional value, it does not reduce the cream layer. Pasteurization kills many organisms, including pathogens, but some putrefactive organism may survive, causing milk to putrefy over a rather time than sourcing. Sterilization involves severe heat treatment to almost completely destroy all microorganisms, resulting to commercially sterile milk with a longer shelf life. So that's all. Thank you for listening.
Hi, good day. I am Rizalgay Columbrillo. In this report, we will explore the significance of microscopic examination in the bacteriologic analysis of milk. Microscopic examination of milk is an important technique in the dairy industry. It involves using a microscope to observe and count bacteria and somatic cells in a raw milk. This analysis provides valuable information about the quality and safety of milk products. By examining milk samples under a microscope, dairy professionals can identify and quantify microorganisms and somatic cells present, which can help assess the milk's overall condition. DMSCC, or direct microscopic somatic cell count, it is a method used to count the actual number of somatic cells in milk. So somatic cells are the cells that make up the body of an organism. So in the dairy industry, the somatic cell count, or SCC, is an important indicator of milk safety and quality. An elevated somatic cell count can indicate the presence of infections such as mastitis. Is an inflammation of the mammary gland in the breast or udder, typically due to bacterial infection via a damaged nipple or teat. Next is the mastitis and the somatic cell count. So mastitis is a common condition in dairy cows where the udder becomes inflamed, usually due to bacterial infection, as what I've said a while ago. So pathogenic bacteria such as Staphylococcus aureus can cause mastitis. When a cow has mastitis, the somatic cell count in her milk increases. So somatic cells, primarily white blood cells known as leukocytes, are part of the cow's immune response to the infection. So therefore, an elevated somatic cell in milk can be an indicator of mastitis and can help identify cows that may require treatment or closer monitoring. Okay, so microscopic procedure. The microscopic procedure for examining milk films was first described in 1910 by Prescott and Reed. It involves using stains to visualize microorganisms and somatic cells in milk samples. The Leibowitz-Weber stain is commonly used for both direct micros- for both direct microscopic count, or DMC, and direct microscopic somatic cell count, or DMSCC. So other stains such as methylene blue, methyl green pyridine E or Y, or ethidium bromide may be used for different milk species. So these stains help to enhance the visibility of microorganisms and somatic cells under the microscope, allowing for accurate counting and identification. So as what you can see in the screen, we have here the method of direct microscopic count. So deliver 0.01 ml of milk on slide and air dry, then stain with Leibowitz Weber stain for 10 minutes. After that, count the bacterial clumps, or the somatic cells with the use of microscope. So in reading the results, the color of bacterial clumps are dark blue. Then in protein, leukocytes, and somatic cells are color blue background, color light blue background, and the fat globules are colorless, and color of the dirt are brown. So we have here we have also here the same sa ginagamit na to sa RBC count. So the here is nag-use sila og 25 squares in counting, then glass slides and cover slip. Okay, so we have here some of the microscopic examination of milk samples. So each figure represents a visual observation made during the microscopic examination of milk samples. So These observations provide insight into the type of microorganisms present and their potential implication for milk quality and safety. So, as what you can see in figure 1, nasa short chains of cocci, then in figure 2, nasa pairs of cocci. So, these observed in milk samples, no? So, the presence of these cocci may indicate poor cooling or dirty equipment. So, if not addressed, these conditions can contribute to bacterial contamination, affecting milk quality and safety. Okay, next, here the figure 3. This figure depicts long chains of Staphylococcus agalactiae, a bacteria known to cause mastitis also. So, the presence of these chains along with their association with somatic cells suggests an active infection in cow that produced the milk sample. Next is in figure 4, illustrates a severe case of S. agalactiae infection from an individual cow. So, okay, next, figure 5. This figure shows Micrococcus bacteria in cluster formation, which may be associated with persistent soils on milk equipment. So, the presence of these bacteria suggests inadequate cleaning and sanitation practices, which can contribute to contamination and compromise milk quality. Next, in figure 6, this figure represents a Pseudomonas strain isolated from raw milk that was stored at marginal refrigeration temperatures for an extended period. So, improper storage conditions can promote bacterial growth, leading to potential spoilage and a decline in milk quality. Next, so figure 7. So this figure displays a gram-positive bacillus strain that exhibits similarities to gram-negative rods. So this observation suggests the presence of the presence of specific bacterial species that should be identified and monitored due to their potential impact on milk quality and safety. So next is in figure 8. This figure shows a spore stain of gram-positive bacillus stain, revealing both vegetative growth and spores. So spores are a dormant form of bacteria that can survive harsh conditions and resist disinfection. So the presence of spore-forming bacteria requires appropriate control measures to prevent contamination and maintain milk quality. Next, next, in figure 9, this figure presents a milk smear showing budding yeast and mold filaments, so indicating the presence of fungal contaminants. Fungal growth in milk can lead to spoilage and affect product quality, so proper hygiene and storage practices are necessary to prevent fungal contamination. Next, in figure 10, this figure displays a gram stain of Prototheca, a genus of algae-like organisms isolated from the milk of cow with mastitis. So Prototheca infection can cause mastitis and impact milk production. Identifying and addressing such infections is crucial for maintaining milk quality and other health. So
I'm Latifa A. Abdulatif and I will be reporting the method of analysis. In addition to the appropriate amounts of carbohydrates, proteins, and fats, breast milk provides vitamins, minerals, digestive enzymes, and hormones. So breast milk also contains antibodies and lymphocytes from the mother that helps the baby resist in any infections. So the milk sample taken from a randomly selected pooled batch using aseptic technique should be sent immediately to the laboratory. So some aliquots of raw milk are the pre-pasteurization for microbiologic testing, and a 10 microliter sample is taken from a randomly selected aliquot from the pooled batch. The method would be the streaking pattern for inoculating media for semi-quantitative bacterial colony counts. A 10 microliter disposable inoculating loop calibrated to contain 0.01 ml of milk is immersed into the milk sample. The loop is then carefully removed and the entire volume delivered to the surface of an agar plate by making a single streak across the center. So the inoculum is spread evenly at right angles to the primary streak, starting at the center of the medium. Then the plate is turned 1800 and the inoculum is spread to cover the entire surface. The next method would be the streaking pattern on blood and McConkey agar plates using a 10 microliters inoculating loop. The primary inoculation, streak at right angles to the 1 degrees inoculation starting at the center. Then plate is turned 180 degrees, streaking evenly to the cover entire surface. So blood agar plate and McConkey agar plate inoculated with 0.01 ml raw milk sample and incubated in 24 hours. So the number of bacteria is estimated by counting the following of colonies that appear on the surface of the media. If a 10 microliters or 0.01 ml loop had been used, multiply the number of colonies by 100 to get the colony-forming units of bacteria per ml of milk sample. And if a 1 microliter or 0.01 ml loop had been used, multiply the number of colonies by 1000 to get the colony-forming units of bacteria per ml of milk sample. Milk suitable for pasteurization should meet the following minimum standards. So if the batch does not meet these standards, the whole batch must be discarded or stored for research purposes. The next method would be the pasteurization. It is a bactericidal and virucidal maintaining microbacterial safety with retention of maximum nutritional and antimicrobial characteristics of human milk, assuring bacteriologic safety. The first one would be the post-pasteurization bacteriologic testing. The first thing you need to do is to remove 1 bottle per batch and get an aliquot of bacteriologic testing. All pasteurized milk should be promptly frozen for storage. Next is any bacteriologic growth is unacceptable for pasteurized donor human milk. If the processed milk does not meet these standards, again, the whole batch must not be dispensed or it can be stored for research purposes. The things and process that you need to follow. The 10 microliter of pasteurized human milk streaked onto a blood agar plate should be incubated in 24 hours at 37 degrees Celsius. After that, we need to examine plates for any growth. Remember that post-pasteurization bacteriologic testing using blood agar plate only for whole or by plate. The next method would be testing for purity of breast milk. This test is randomly done to check for adulterated breast milk. We should use sterile screw cap test tube, 1 ml of donor human milk, 1 ml of 8N solution of sulfuric acid, 8 ml of distilled water. Take note that mix and allow to stand at room temperature for 5 hours. After the method has been done, observe these results. If a precipitate forms in the mixture, the solution is positive for bovine or cow's milk. So the entire donation should be disregarded and donors should be removed from the donors list. Negative for precipitate is the occurrence of breast milk. Positive for precipitate is the occurrence for the cow's milk. That ends my report. Next reporter, please.
Good day to you, Sir Kent. I'm Sarah Asinas, and let's now proceed on the interpretation of results on bacteriologic analysis of the milk. So first is the total bacterial count or standard plate count. So sa TBC or SPC, pag gamay ang iyahang count, which is less than 10,000 colony forming unit per ml, nag-indicate siya that there's a good hygiene and refrigeration practices during milking, processing, and storage, and there's a sign of a fresh, high-quality milk. Next, if is the moderate count, which has 10,000 to 100,000 colony forming unit per ml. This suggests fair practices, but there might be minor issues with handling or refrigeration. Sa high count, that is greater than 100,000 colony forming unit, nag-indicate siya na merong poor hygiene and naay potential issues sa pag-handle sa milk during refrigeration, or pwede pod na ang animals na differential or if they're sick. So, kani nga level contamination can affect the milk quality and its shelf life. Next is the coliform count. So, if there's a low count that is less than 10,000 colony forming unit per ml, good sanitation, and there's a low fecal contamination. If taas naman, that is greater than 100 colony forming unit per ml, nag-suggest na there's a significant fecal contamination and there's a poor sanitation or there's a water contamination. So if taas ang iyahang counts, there's a serious concern that indicates a higher risk of pathogens. And these are some of the presence of specific pathogens: the Salmonella, Listeria monocytogenes, and Escherichia coli. So, the presence of any pathogenic bacteria may indicate a serious health risk. So, this bacteria nagakos siya foodborne illnesses, and it requires immediate action to prevent consumption of the contaminated milk. And then sa somatic cell count, if there's a low somatic cell count that is less than 200,000 cells per ml, this indicates good animal health. So lower counts are often associated with high quality milk. So if gamay ang iyahang somatic cell count, taas ang quality sa milk. So if taas naman ang somatic cell count that is greater than 200,000 cells per ml, nag-suggest siya mastitis. It means this is the inflammation of the mammary gland in dairy cows, which can affect the milk quality. So if taas ang yahang count, nag-indicate siya poor animal health and yahang welfare. So next is the psychrotropic bacteria count. So psychrotropic bacteria count can grow at refrigeration temperatures and nagkakos siyang spoilage. So if there's a low count, it indicates good shelf life and freshness. And if there's a high count naman, there's a potential spoilage issues even if the milk is being refrigerated. And for interpreting results for action, so for producers, regular testing and monitoring of discounts can help identify the issues in animal health, milking practices, and storage conditions. It's crucial for maintaining the quality of milk and ensuring the health of the consumers. And for regulatory bodies, ensuring that milk meets the legal standards for bacterial counts is essential for public health. Actions may include recalls, fines, or other regulatory measures for products that do not comply. And for consumers, awareness of these factors can guide choices and emphasize the importance of purchasing milk from reputable sources. So there's other results also that if there's a precipitation na nag-form, during the mixture of the solution that is positive for bovines or cow's milk. So the entire donation should be and must be discarded, and donor should be removed from the donor list. So, if negative ang iyahang precipitation, it is for breast milk, and for cow's milk, positive ang iyahang precipitation. And to conclude, bacteriological analysis of milk provides essential information about its safety, quality, and shelf life. Interpreting these results can help in making informed decisions and to ensure the health and satisfaction of consumers, as well as the adherence to regulatory standards. So now let's proceed to the next.
So good day, everyone. I'm Ivy Dintonish. I will be reporting the pros and cons of bacteriologic analysis of milk. So for the pros of bacteriologic analysis of milk are the following. First, the quality assurance. Bacteriologic analysis of milk ensures quality assurance by detecting the presence of harmful bacteria such as Salmonella, Listeria, and E. coli, which can pose serious health risks to consumers. Second is public health protection. By identifying and quantifying bacterial contamination, bacteriology analysis helps to protect public health by preventing the spread of foodborne illnesses associated with consuming contaminated milk. Third is regulatory compliance. Many regulatory bodies mandate bacteriologic analysis of milk to ensure compliance with food safety standards and regulation, thereby promoting consumers' confidence in dairy products. And next is early detection of spoilage. Bacteriologic analysis can detect spoilage bacteria in milk, enabling dairy producers to take corrective actions promptly to prevent further deterioration of milk quality and ensure product freshness. Lastly, process optimization. Regular monitoring of bacterial counts in milk through bacteriology analysis allows dairy farms and processing facilities to optimize their production processes, minimizing the risk of contamination and improving overall product quality. And for the cons naman of bacteriologic analysis of milk are the following. First is time and cost. Bacteriologic analysis of milk requires specialized equipment, trained personnel, and dedicated laboratory facilities, which can be time-consuming and costly for dairy producers, especially small-scale operations. Second is complexity of interpretation. Interpreting bacteriologic analysis results requires expertise in microbiology, as the presence of certain bacteria may not always indicate contamination or spoilage, leading to potential misinterpretation of result. Third is limited scope. Bacteriologic analysis primarily focuses on bacterial contaminants and may overlook other potential hazards such as chemical residues or allergens, necessitating the use of complementary testing methods for comprehensive food safety assessments. And next is sampling variability. Sampling variability can impact the accuracy of bacteriology analysis results, as microbial population in milk can vary depending on factors such as herd management practices, environmental conditions, and sampling techniques. So lastly, the false positive or false negative bacteriology analysis may yield false positive or false negative result due to factors such as sample handling errors, cross-contamination, or the presence of inhibitory substances. leading to potential misdiagnosis of milk quality. So, in conclusion, while bacteriologic analysis of milk offers numerous benefits in terms of quality assurance, public health protection, and regulatory compliance, it also presents challenges such as time and cost constraints, complexity of interpretation, and limitation in scope and accuracy. To maximize the effectiveness of bacteriologic analysis, dairy producers should implement robust sampling protocols, invest in training for personnel, and integrate complementary testing methods for comprehensive food safety management approach. So that's all for the pros and cons of bacteriologic analysis of milk.
Good day, everyone. My name is Kerdy Tihada from BSMNS 3C. Today, I am going to discuss comparison between bacteriologic analysis of water and milk. Bacteriological analysis of water and milk serves similar purposes in assessing microbial quality and safety, but there are notable differences in the methods and considerations involved due to the distinct characteristics of these two liquid matrices. Here's a comparison between bacteriological analysis of water and milk. Sample collection and characteristics. Water samples are collected from natural sources such as rivers, lakes, or groundwater, as well as from treated water supplies. These samples vary widely in composition and can contain a diverse range of microorganisms, including bacteria, viruses, and protozoa. For milk samples, they are obtained from dairy farms, processing plants, or retail outlets. Milk is a nutrient-rich liquid with a complex composition, making it an ideal medium for microbial growth. The primary concern in milk is the presence of bacteria, including spoilage organisms and pathogens. Transportation and preservation. Water samples need to be transported to the laboratory quickly and under controlled conditions to prevent changes in microbial populations. Preservation techniques such as refrigeration or addition of chemical preservatives may be necessary to maintain sample integrity. Milk samples require rapid transportation and careful handling to prevent microbial growth and spoilage. Cooling is essential to inhibit proliferation of bacteria, particularly psychotropic bacteria, which can thrive at refrigeration temperatures. Sample preparation. Water samples may require filtration, dilution, or enrichment steps to concentrate bacteria and remove interfering substances before analysis, while milk samples may undergo homogenization and dilution to ensure uniform distribution of microorganisms. Also, to obtain representative samples for testing. Homogenization disrupts fat globules and ensures even distribution of bacteria throughout the sample. Analytical methods. Traditional bacteriological analysis for water often involves culturing bacteria on selective and differential media. Common techniques include membrane filtration, multiple tube fermentation, and spread plate method. Membrane filtration. The water sample is passed through a filter membrane with a defined pore size. The membrane is then passed onto a suitable agar medium and colonies that develop are counted, while multiple tube fermentation or MPN is a method which involves a serial dilution of the water sample followed by inoculation into tubes containing selective growth media. The presence of bacteria is determined based on gas production or other observable indicators. Spread plate method, on the other hand, is a known volume of water sample spread onto the surface of a solid agar medium. After incubation, colonies that develop are counted and used to estimate bacterial counts. Additionally, molecular methods may also be used for the detection of specific pathogens in water. One of the primary analyses conducted in milk is the enumeration of total bacterial count or TBC. This is used to assess the overall microbial load in the milk sample. This is typically done using agar plate count methods such as the standard plate count or SPC technique. In this method, serial dilutions of the milk sample are plated onto agar plates, and after incubation, colonies are counted to estimate the number of viable bacteria present. Regulatory standards for water quality are typically based on the presence of indicator organisms such as coliform bacteria E. coli and the enterococci, as well as specific pathogens. Standards vary depending on the intended use of the water, such as drinking water, recreational water, or agricultural water. For milk, regulatory standards for milk quality focus on bacterial counts, somatic cell counts, and the absence of specific pathogens. These standards are designed to ensure the safety and quality of dairy products for human consumption. Overall, while both bacteriological analyses share common objectives of ensuring microbial safety and quality, the methods and considerations are tailored to the unique characteristics of water and milk as matrices. That would be all. Thank you all so much for listening. Again, this is Kerdi Tihada reporting the comparison between bacteriologic analysis of water and milk.